Hi everyone. Today we're going to explore the human immune system. Here we have the common cell.
Hi, I'm just your average cell. They call me macrophage. I live in your body.
Oh no, that looks like a virus.
Hee. I am a virus. I want to cause some trouble.
No, please don't come near me. I don't want any trouble today.
Too bad. Tag. You're infected. Mwahaha.
Oh no.
This is what occurs when a common cell undergoes phagocytosis, bringing the virus inside the cell by engulfing it.
Oh no, this virus will kill me if I let it stay inside. Enzymes, help me out.
Da da da!
Slash. The enzymes cut the virus.
Ah, you're hurting me.
Antigen fragments are formed and float inside the cytoplasm of a cell until they are picked up by the MHC inside the cell.
Oh, that's my cue. Clean up, everyone, everywhere, clean up. Cell membrane is over there.
Goodbye, cruel world.
The class 1 MHC molecule in the cell brings the antigen fragments to the membrane of the cell for what is called antigen presenting.
Now my chore is done. Yippee.
Well, gee, look how fun these blocks look. Just one quick touch should be okay. Ooh, that was fun. Ooh, look, another one. Ah, it stuck to me.
Sorry, bro, it's my job. I've got two chains, the A chain and B chain linked by disulfide bridges that form the binding site.
A cell has both self-antigens and non-self antigens. Upper T cells and killer T cells will target non-self antigens and cause death to those cells presenting non-self antigens. They will typically leave self-antigens untouched. A protein called CD4, which exists on the surface of T cells, binds to the MHC molecule and helps keep the helper T cell and the antigen-presenting cell together. This link creates a cell response, as you soon shall see with the release of cytokines.
Wait, dude, what are you leaking?
The great cytokines have come to save the day. The common cell and all the body will be saved.
Phew, I don't know how much longer I could have stayed cramped inside that cell. I'm going to take a journey around town to check out what I've been missing.
The activation of helper T sends a signal to the cell to secrete cytokines. Cytokines, specifically IL-2, serve to stimulate more helper Ts, but also stimulate killer Ts.
Don't test me. I will annihilate you, punk. Any virus tries to step in my way, you better watch his back.
The big bad killer T's come across the cell with the specific antigen receptors he's looking for. Meanwhile, he sends his sidekick, the memory T, to the side to watch.
Yo, Wimp, I got a bone to pick with you.
The result of the killer T's grabbing the receptors is catastrophic. The common cell falls and disintegrates to pieces, all else falling to chaos.
What really happened was when the killer T cells interacted with the receptors, they elicited a signal to create poison in the cell that caused the rupture and the death of the infected cell. This released the antigen fragments into the surrounding environment to be picked up by antibodies and destroyed fully. Killer T cell binding to the receptors facilitated by the protein CD8.
Well, there you have it, kids. Once again, the immune system comes to save the day. Don't forget to thank your T cells next time you get an infection. Uh-oh, what's that?
I'm back. Ha! I bet you thought you'd seen the last of me.
I remember you. I got this. Hey, back off. I beat you before. I'll beat you again.
When the virus comes around again, the immune system has an easier time fighting it because of the memory T cells. These cells, which are put aside in the earlier fight against the infection, recognize the specific infection from memory and are activated when it appears inside the system. The end.
All right, don't sit down, please. Everybody just kind of line up in front because we're going to do a Q&A really quickly. Or maybe not so quickly. All right, so in the audience, was there anything that you see on the actual list of events that you think that they did wrong or is missing or you have questions about? Yeah, Sammy.
For the last one, it says the infected cell lyses, but I think you said something about like a protein coming in and like making the cell die. Is that like the same thing?
So when a cell lyses, we said that the cell ruptures and dies, which is the same thing as lysis. Hence the jacket coming off when I fell. I don't know if you could see it from where you were since I was on the floor up here. But the cell ruptures and dies because of the proteins secreted by the killer T cell.
Yes, Howard.
Can you explain step 7 again? What's the IL-2? What's the TH cell?
Oh, I can explain that one. So, the IL-2 is, the IL-2 is the specific kind of cytokines that are released that help stimulate like more helper T's to be made, and also like stimulate the activation of the killer T's. So it's actually needed to start that process.
Okay, one question I had, it was just a little tiny thing. You used the example of the virus in this case, but then you had the virus infecting the macrophage itself. So typically what happens is the macrophage will eat cellular invaders, like, there's a bacteria cell, it'll go ahead and eat that. What happens with viruses is we don't really typically respond to those until they've infected our own cells. And so they'll infect just one of our normal body cells, and then that will lead our own body cells to display non-self antigens, which will then be a signal to kick off everything that you guys talked about there. So typically, there's always an exception, but typically the viruses won't actually infect the macrophage, and that's what sets off the whole pathway. You can still set off this whole cell-mediated pathway with a macrophage, but just not with the example of a virus. So that was just a little tiny thing. Everything after that point was totally right, but it was just that little tiny thing at the beginning that I noticed that didn't quite follow what it was supposed to be doing here.
We had the name of the macrophage to be just like common cell, right?
Yeah, common cell. And then the other thing that I liked, but I think needed a little more detail, was Theodore at the end. He was the memory T cell, and he just came along with a lightsaber and just destroyed Posen, who was a virus, directly. Now, in reality, what really happens there? So remember what I said before. If the virus comes back, the same virus, it has to be the exact same virus, right? The exact same virus comes back, it infects body cells. Your body cells present antigens. Your own body cells, the MHCs, present the antigens to the outside of the membrane. Now the memory T cell, does it kill the virus directly?
Well, the memory T cell already has the correct antigen receptors to the antigen that's within the virus, so you don't have to go through the whole process of making those specific killer T's again, so you can go straight to destroying.
Right. So the memory cell proliferates and becomes that exact same specific killer T that can identify that antigen on that specific body cell and kill only those body cells with those antigens. So you don't have to go through the whole process of figuring it out, because he's got the exact gene expression, the exact genes necessary to go ahead and identify that particular antigen. Now, before we go into the antibody-mediated group, one thing you also have to understand is that when we're talking about immune cells, we know they divide by mitosis, right? Typically, that would create clone cells. But what T cells, and actually to more an extent B cells, do is they have something called somatic gene rearrangement. There's gene shuffling. There's actually a high rate of mutation. And so you get this great diversity in B cells and T cells, which allows all these cells to have all these different receptors to try to figure out the invader. And then once the invader's figured out, then we see the whole process of what we call amplification. Like, okay, once that lock and key thing is figured out, then that specific cell starts multiplying and starting out sending chemical signaling to make other cells that can fit that particular antigen start multiplying, and then that's why you get this immune response that happens so fast. And that part would be positive feedback. Anytime when you hear the word amplification, that would be the positive feedback loop. Now, what other questions do we have from the audience here? And remember, your test is coming up Thursday, so you've got to be able to explain this pathway, understand antigen, MHC, all that good stuff. IL-2 is just interleukin. That's a type of cytokine, a specific type of cytokine. Perforin's a chemical that causes the cell to lyse. A special type of protein that causes the cell to lyse. Cytotoxic T is another word for killer T. Yeah, Seth.
What does CD8 refer to?
Oh, so we said that the killer T binding to the receptor is facilitated by the protein CD8. So it's just, yeah.
So a little more detail on that. When you have an MHC presenting an antigen, then there's another cell that has to come along and recognize that, right? There has to be a receptor there to recognize. And then along with that, there's this thing called either CD8 in this case or CD4 that helps with the binding to make that complex come together, to get that recognition, to get that to dock. And then you've got also the cytokine communication pathways that cause amplification or recruit other molecules to come by. So let's get back to the helper T here for a second. So the helper T simulates the cytotoxic T, and that makes them go ahead and identify those particular pathogens and kill those pathogens. But then the helper T is also releasing cytokines that will actually lead to its own amplification. So it's kind of almost a little positive feedback loop there. But then the cytokines also go over to the antibody-mediated pathway. So the helper T is like the central general of the entire immune system. It is a part of this cell-mediated pathway, and it's a part of the next one we're going to talk about, the antibody-mediated pathway. And so both of these processes, one doesn't happen before the other. They're both happening at the same time in a coordinated way to get rid of the invader. Now, what other questions do we have from the audience? Oscar? Do you want to ask anybody to prom right now, since this will go on YouTube?
Coffee.
Any other questions? Jessica's getting really red. Come on, you're going to be stars. Come on, Amon.
Like virus.
Come on, Mom, we'll make you a meme.
Mr. Erdmann, I hope you enjoyed my meme.
I do enjoy your memes that you post on the Facebook group. So that's it. We've got this. Okay.